All right, good morning, everybody. I started recording this lecture like half an hour ago, and I was so tired that I just was not talking well. So I decided to start it over for all of our sakes. Okay, Mendelian genetics. So this idea is Mendelian genetics is commonly used to predict phenotypes, like how you inherit phenotypes over generations and generations, or genotypes over generations and generations. But it really all started with this guy named Mendel. And so Mendel lived about the same time as Darwin, 1820s. The dude was super smart, and so he didn't come from a really privileged family, but his parents had enough money that when a professor identified him as being like unique and gifted, if you will, that his parents were like, okay, we need to like bend and send this kid to grad school. So they kind of pulled from their life savings and sent their brilliant son to grad school. Now, the problem with really smart people is that when they learn a lot about the world and they realize that they know a lot more than the people around them, or, they tend to get depressed. And so um, Mendel battled depression. He maybe didn't feel a sense of connectedness to those around him because he was so brilliant. And so just like most people that go through these cycles of like, I learn some information about the world, I'm not sure how to deal with it, I'm going to go through depression. They typically find religion in some form or another. Nowadays, it's kind of this like what is it? Like, I can't even think of the name of it. Anyway, so he found religion for the 1820s, which was basically the Catholic Church. And so what's interesting about the Catholic Church is that during this time period, there was a belief that science would be able to elucidate truths about the world, about life, that would support the Catholic mission. Because if God is truth and science can find truth, and science is part of religion, like they were kind of the same idea. Nietzsche recognized what was happening, and it was that the more phenomena that science was investigating through these Catholic monastery labs, the more, or I should say, the less they needed a spiritual explanation for the phenomenon. And so he was watching these monasteries slowly kill off the religion by doing science. And so that's why he famously declared God is dead. It wasn't because he like literally thought God was dead. It was like, no, he's like, I see where this is going, and I can see that it will not have a good outcome in the 20th century. And so if you look forward, right, Nietzsche was in the 1800s. He predicted the 20th century would be the bloodiest century on record in human history. And think about what happened in the 20th century: World War One, World War Two, Korean War. I mean, Vietnam, all this Desert Storm, and that's just American conflicts. So you got Maoist China, you got Stalinist Russia. I mean, Nietzsche was another Prussian guy, just like Mendel, super freaking smart. And he was able to see, like, I see exactly what's happening. We're starting to slowly shift our shift our belief structure away from the phenomena of religion towards like the tools of science. And what he was realizing was that some of the morality, like the personal morals and ethics that were ingrained in the religious doctrines, would not make the transition into the scientific objective landscape, and that it would cause like a lot of evil stuff to go down in the 20th century. And he was right. So I digress. Anyways, okay. So his monastery was lab. So he was like, cool, I get to do some research while I'm here. I'm smart. I love to do that kind of stuff. So that's that's what he did. And what he did was he was literally trying to he knew about Darwin. He knew about Darwin's proposals for natural selection. He knew there was a whole community surrounded Darwin. It wasn't like Darwin is at the lead of it; he was just a part of it. And that whole community, what they believed was like, okay, we can see from the fossil record, we can see from geology, we can see that the Earth actually appears to be older than 6,000 years. Then they could also see that there were species that were dead, like that we could find bones. There's their bones, but but we couldn't place them like with any living species. And so what it suggested with it was that species changed over gradually over long periods of time. And that was pretty well established before Darwin came along. And Darwin, all he did was he codified it in a mechanism, and that mechanism was natural selection, which we get to in a few lectures. And so Mendel knew about this whole community and what they were trying to do. And he was like, no. I'm going to prove you guys wrong. I'm going to show you that traits are discrete, and they are not blended together in the phenotype like Darwin was suggesting. Okay, so what Mendel did was he used hybridized pea plants because pea plants are relatively quick to breed, like they have short generations, and they have a lot of dimorphic traits. And so dimorphic traits would be like if you walked out in the world and everyone was either blonde hair or brown hair. That's not true. Or everyone was either brown-eyed or blue-eyed. And pea plants had these discrete dimorphic traits that he was able to say, okay, I can use these in my experiments to show that even though it appears traits disappear in hybrids, they will reappear later on down the road in future generations. And so what he was basically suggesting was that things will appear to change in the short term, but in the long term, the old parental phenotypes that once looked like they disappeared will show back up again. And because he had so much time on his hands and because he was funded by the church, he was able to do thousands and thousands and thousands of these experiments and these crosses and these hybridizations. And so he was able to predict or use mathematical models to be able to predict the transmission and expression of these phenotypes in the future generations. And so these rules that he came up with are still used today and still taught in our science labs and rooms, which is great because that's what we're learning about now. So he came up with this idea of the law of segregation. Now, in his time period, they didn't know about alleles or genotypes or anything like that, but he sorry, that was my wife taking off for the dentist. Okay, so he had this law of segregation, and he didn't know about genotypes, but you can assume he was able to make an inference about what we now call alleles. Is he was like, there are traits that get boiled down into these little packets, these discrete packets that can be passed on to offspring, and each parent is going to pass on one of these discrete packets for a trait. And then the two discrete packets that the offspring receives, if one of them is dominant, it will mask the effect of the other one. He was able to see this. Now, he didn't know what those packets were that were getting passed on, but he was able to identify them and identify how they were their modes of transmission. And so his law of segregation was basically, okay, if you have a parent that's passing on a packet, right, that has two packets and they're going to pass on one of them, right? Let's say dad has the genotype big B, little b. So he's got a B packet, a big B packet, and a little b packet. He's going to pass on either the big B packet or the little b packet. He's not going to blend these two B's together and make some blend and pass that on. No, he's going to pass them on discretely. The law of independent assortment is this idea that, okay, if I take the law of segregation, that there's these individuated packets that can be passed on, and I take that for more than one trait. So I say I got my B packets over here, I've got my A packets over here. Whatever I pass on for my B packets, whether it's the big B or the little b, makes no effect on whether I'm passing on the big A or the little a on my other packet. And that's the law of independent assortment. So dad has a heterozygous genotype for two traits. If he passes on the big A, he still has an equal chance of passing on big B or little b. They're independent of each other when they get passed on. Now, I have to give you a caveat just because we're not going to talk about this a whole lot, but I want you to consider something. So you guys already went through mitosis and meiosis. We didn't do it in the lecture format, but you did it in the lab. In which, if you remember, right, during meiosis, there was a crossing over that happened. So there was a chance for certain alleles to cross over and land on another chromosome with an allele that it previously wasn't on the same chromosome as, blah, 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 right? So alleles can shift around a little bit. This law of independent assortment works just fine as long as the alleles, like the two traits, like the A and B trait, are not close together on the chromosome. Because this idea of like, okay, there's like a chunk of the chromosome that gets transferred or crossed over. If that chunk of chromosome that's getting crossed over from one to the other has the two traits on that chunk that's getting transferred, well, then they don't independently assort. But as long as there's enough distance between them, they will independently assort. And so that's my caveat. It's not on the test. It's just if you happen to do this, if you happen to move on to genetics or something, you'll definitely deal with this idea of traits being stuck together if they're close on the chromosome when they get trans. Okay, so my kid's awake now. It's 6:30 in the morning. Okay, so moving on, let's look at the law of segregation, but from a visual standpoint. Okay, So if we take this pure breeding population of purple flowers, that means if I have if I'm Mendel, right, and I'm breeding flowers together, that means in one greenhouse I have only purple flowers, and every time I cross those purple flowers together, I only get purple flowered offspring. That's a pure breeding population of purple flowers. That is the P generation. That's this one. And then in a separate greenhouse, I have a population of white flowers. And then the same thing, if I breed those white flowers together, I only get white flowers. Now, for the white flowers, it's easier to understand or easier to see that it's a pure breeding population because there's no masking going on, right? If a dominant purple allele shows up in the white greenhouse, it's going to be a purple flower and it's going to stick out like a sore thumb. The purple flowers might be able to hide the white, like if they had a white allele in there a little better, but eventually they would, at some point, the two alleles would get paired up. The two recessive alleles would get paired together, and you'd have a white flower show up in the purple greenhouse, and you'd be like, crap, I have contamination. But anyways, considering that these two come from different greenhouses and they're kept isolated, you have a pure breeding population of purple flowers, so that would be two capital P's, and a pure breeding population of white flowers, two lowercase p's. They each, right, the law of segregation, they each pass on one of their P's. So the pure breeding purples can only pass on a big P. The pure breeding whites can only pass on a little p. And then together they make a heterozygous offspring, these purple flowered offsprings. Okay. The offspring, like we said, the F1 generation. So anytime that you have a parent generation that makes offspring, we call the offspring generation the F1 generation. And they're all purple. They have a purple phenotype. It would appear like the white flower just disappeared. Now, when we cross the F1s, meaning we take the F1 flowers and we cross the F1s together, so when we cross the hybrids together, what we see is sure, the offspring, a lot of them are purple, right? Just like the parent, just like the grandparent. But occasionally that white phenotype shows back up. It skips a generation. And so that demonstrates Mendel's law of segregation, that the alleles never blended together. That white flowered allele never went away. It never blended into the purple and disappeared. It was always kept discrete by itself and was passed on in a discrete way because it showed up in a 3-to-1 ratio in that F2 generation. And so the F2s are always the grandchildren of the P generation. Now, the law of independent assortment, we have to look at two traits in order for this one to work. And so the two traits that we're going to look at is yellow, the color of the seed pod, and then also the shape of the seed pod. So whether it's round or wrinkled. Now, the yellow is dominant, and that's coded for by a capital Y. The round is dominant, and it's coded for by a capital R. So if we take, and imagine these happening in two separate greenhouses. So we take our yellow round pea pod greenhouse and our green wrinkled greenhouse, and we take pollen from each and cross pollinate them, then the individual that's produced is a hybrid of the two parents from the two greenhouses. Now, if you consider this, the parents of the P generation, so the P generation, can only pass on a capital Y and a capital R together and a lowercase y and a lowercase r together in their gametes, respectively. And so what you end up with in the F1 hybrids is you end up with individuals that have one capital Y, one lowercase y, one capital R, one lowercase r. And what I did was I color coordinated these so you can see what happens with the law of independent assortment. Because the capital Y and the capital R are on the same chromosome. They're in the same gamete. They're coming over from one parent. Same with this lowercase y and lowercase r. And so you could consider them as like this capital Y and this capital R are on one chromosome, one set of chromosomes, just like from our last lecture. And the lowercase y and lowercase r are on the other set of chromosomes, making one set of homologous chromosomes for that one chromosome. So anyways, the F1 hybrids are all going to be yellow and round because they're going to have one dominant allele from each. But when we take the hybrids and we blend them together, this is where it gets a little bit tricky. Now, these individuals, due to the law of independent assortment, the individuals now can pass on a capital Y and a capital R together, just like the grandparent did. But because you have the crossing over in meiosis, now they can also pass on a lowercase y with a capital R. That's the law of independent assortment. That was once in separate grandparents can now become in one chromosome. So each grandparent, right, the yellow round one and the green wrinkled one, each grandparent had separate traits on separate chromosomes. They come into the hybrid individual, and that hybrid individual, through the meiosis, through crossing over, can now exchange some of the alleles between the chromosomes and end up with one chromosome that has a dominant allele and a recessive allele from the other grandparent. So it's like taking the two traits from the grandparents and putting them on one chromosome. And now they can do that with equal chance. And that's the law of independent assortment. So the F1 generation has an equal chance of passing on a capital Y and a capital R, a lowercase y and a capital R, an uppercase Y and a lowercase r, or two lowercase y and r. And so what you can notice here is that the lowercase y paired with the dominant r illustrates that independent assortment. That's what we just talked about. Same thing with the uppercase y and the lowercase r. And then when you blend them together in the F2 generation, so this matrix here, this grid of 16 squares, represents all of the possible F2 generations phenotypes. And what we see is in this dihybrid cross, the offspring's phenotypes show up in a 9:3 to 3:1 ratio. The 9 being the dominant, so that would be the yellow round. The 3 showing up is split, so that means that it's recessive phenotype for one of the traits, yet dominant for the other. So in this case, the 3 round green ones are recessive for the green color, but dominant for the roundness. And then we go to the next one, and that one will be 3 that are dominant for one, but recessive for the other. So they're 3 wrinkled, but also 3 yellow at the same time. And then finally, that last one will be the recessive for both traits. It'll be green and wrinkled. And that's the law of independent assortment. All right, so let's get to practicing Mendelian genetics. So let's say that hair color in hamster is controlled by one gene with 2 alleles. So you got a big B allele codes for black fur, and it's dominant. So anytime a hamster has a big B allele, it'll be black. The little b is recessive, and it codes for white fur. So let's say we have a hamster with the genotype big B, so a pure breeding hamster from a black furred population has offspring with a hamster with the genotype little b. Well, what color of fur will the offspring have? Let's figure this out. So we've got our black hamster over here who's homozygous dominant, our white hamster who's homozygous recessive, and what kind of offspring are they going to make? Let's take a look. Well, let's take all the possible gametes that one hamster can make. So we've got the black hamster and the white hamster, and then you place the gametes into their own cells along the border of this matrix, of this Punnett square. So the black hamster will only be able to pass on a big B, and the white hamster will only be able to pass on a little b. Each one of these squares around the border represents either sperm or egg. It depends on what the gender of the hamster is we're looking at. Considering that this white hamster is the female, well, then that would be an egg with a little b allele in it, and another egg down here, a separate egg with a little b allele in it. And this hamster up here is making a separate sperm with the big B allele and another separate sperm with the big B allele. So what we can do then is we can see, okay, what are all the possible combinations for the offspring? So we fill in the table down here, and what we see is that all of the offspring from that cross will have to be heterozygous, right? They will all have black fur from that cross. Okay, so when we get to test crosses, this is where we have somebody that's expressing the dominant phenotype. So in this case, it could be you go to a pet store and you buy a black hamster from the pet store for breeding, right? Maybe you want to make more black hamsters, right? And you say, well, okay, what's its genotype? It's a black hamster, but what's its genotype? Is it big B, or is it big B, little b? How do you know? You look at it, but you cannot tell. The only way that you can tell is if you do a test cross. And so ignore the sheep, imagine they're hamsters. It's the exact same situation, okay? So if you have a black hamster, you don't know whether it's big B, or big B, little b. So what you do is you cross it with a known homozygous recessive, or a white hamster. So we bought this black hamster from the pet shop. We don't know what its genotype is. We make it have babies with a white hamster, and here's how that works out: is that if the offspring come out mixed, meaning that some of them are white and some of them are black, then you know that the genotype of your black hamster was heterozygous if they're mixed. If the offspring are all black fur, then you know that the genotype of the original parent was homozygous dominant. So if he was homozygous dominant, right, that means he can only pass on a big B allele. Oops, back up. Big B allele. He crossed it with the recessive white hamster, and they can only pass on the little B allele. So if the unknown phenotype or the unknown genotype from the dominant phenotype, it's like get tongue tied here. If the unknown genotype from the known dominant phenotype is homozygous dominant, so if that individual has two big B alleles, it can only pass on the big B allele. So the offspring are, of course, all going to be black. But if the unknown genotype is heterozygous, half of the time he's going to pass on the little B allele. 100% of the time, the white hamster you breed him with is also going to pass on a little B allele. And so the offspring should be 50-50-ish. I mean, it's always a chance, right? But the offspring produced from that will be a mix of white and black hamsters. And so that's how the test cross works. So that's how test cross work. Okay, types of crosses. So we have two different types of crosses. A monohybrid cross is where you take just one trait. So that could be like eye color, hair color, and you look at how that one trait is transmitted through breeding. So an example of this would be like you have a B trait, right? Maybe it codes for eye color. You cross big B with big B, little b. That would be a monohybrid cross. A dihybrid cross is where you look at two traits and you trace them through breeding. Typically, when we talk about hybrid crosses, we're talking about crossing two heterozygous individuals together. But you can look at it like mono is just looking at one trait, dihybrid's looking at two traits. So dihybrid's where you cross two traits through breeding. So maybe you take an individual with big A, big B, little b, and big A, and little b, and say, okay, what are all the possible types of eye colors and hair colors that those two individuals could pass on? And now in a dihybrid cross, one parent is going to have the dominant genotype for the two traits, big Y, big R, and the other one's going to have the recessive genotypes. When you breed them together, you get a hybrid, and then you get the F1, you cross the F1 generations together, and you make the F2 generations, and the F2 generation has a 9-3 to 3-1 ratio of phenotypes, which we talked about in the last part. Okay, non-Mendelian inherited traits. So, although most genes are carried on the most genes are carried in the autosomal chromosomes, so we'll take a look at those. So the autosomes are traits that are on homologous chromosomes 1 through 22. So I'll show you what those are in just a second. All humans are going to be diploid, male or female. They're going to be diploid for those traits and have two alleles for each gene. So it follows the Mendelian patterns of inheritance as long as it's just one gene is one trait. So I circled here on your slide, I circled here all of the autosomes. So you can take a look at that. That's all 22 of the autosomal chromosomes. Notice there's one we left out, which is the sex chromosomes. All right, so then we have the sex-linked types of traits, which are carried on the X chromosome. But they are missing from the Y. So if you have a trait that's found on the X that's also found on the Y, then it's going to behave just like an autosomal chromosome, and it's going to follow the Mendelian patterns of inheritance. But in the case that you have traits that are missing from the Y but are found on the X, then females, which have two X chromosomes, will be diploid, but males are haploid for those traits. And don't worry, I'll show you a picture of this on the next slide. And these do not follow your typical Mendelian patterns of inheritance. And that's just these sex chromosomes, so X and Y. So let's take a look at the X and Y chromosomes. In females, they have two X chromosomes, and in males, they have an X chromosome and a Y. Now, the Y chromosome has a lot of the same genes as the X chromosome. The problem with the Y is that it's shorter. So females have two X, males have an X and Y. The Y chromosome is shorter, and so it's missing some of the genes that are found on the X chromosome. And that's why males who are XY will be haploid. They'll only have one copy of those genes that are found on the X. That's a problem because if one of those genes is non-functional or bad, that male will have that disease, that recessive disease caused by that dysfunctional allele. So let's look at how these things are inherited. Males have an X and a Y. Females have two Ys. So females pass on two copies of their X. Males pass on a copy of the X or a copy of the Y. And we can put them in a Mendelian or in a Punnett square and see how these things are passed on. And what we see is that half of the children produced by a man and a woman, half of them will have two X chromosomes and half of them will have an X and a Y. And so that's why 50% of children are boys and 50% are girls. So here's a way that we can look at X-linked traits. So let's break this down for a second because it takes a second. Just kind of take in all this information. Okay. Here we have a mother, and she's unaffected, but she carries this blue, we could call it a mutation, we could call it a recessive allele on her X chromosome. So she has this bad allele, if you will, on one of her X chromosomes. But because she has a good copy of it, the green copy, she's unaffected. See how she's a light gray? Now the dad, he's got two good copies. He's good to go. Okay. He's unaffected. He's light gray. The daughter they produce, so the mom will pass on her affected X chromosome at some point, and the dad will pass on his X chromosome that it is affected, but the daughter is not going to have any problems because she has a good copy that she got from mom. Now, the son, though, will get the Y from dad, but he'll get the bad copy from mom right here, and the son will be affected because he only has that one copy, and it's a bad copy, and so he has the disease. And then the two daughters, the daughter and the son that are unaffected, are easy to trace because the dad passes on an unaffected X, the mom passes on her unaffected X, and the daughter's fine. Same thing for the son. Dad passes on his Y, mom passes on her unaffected X, and the son is okay. So half the boys of a carrier mother will be affected. That's how sex-linked traits works. And so only if the male inherits the defective copy, he will have the disease. And then so females, in the case of X-linked traits, are typically more often than not carriers. You know, they're usually less affected by these sorts of things. So I'll give you an example of a sex-linked trait called hemophilia. Now, hemophilia is one of those things where it's a terrible disease to have. It's found mostly in British royalty. And it's this inability to produce platelets. And platelets are what clot when you get a cut or when blood vessels are damaged. And so affected individuals that have hemophilia can bleed out very easily because they can't clot. An affected X chromosome will be marked with, sorry, I kind of messed that up there. It's not the affected X chromosome, but the X chromosome will be marked with either a superscript H or superscript little h, capital H or little h. The little h, the lowercase h, is going to be the one that's dysfunctional. That's going to be the one that carries for hemophilia or bad platelets, codes for bad platelets. Okay. So capital H codes for normal, little h codes for no platelets. Now, notice all the males only have one copy of the X. So here's my males, X and Y. They only have one copy of the X, which makes them more susceptible to having hemophilia. So hemophilia affects males more than females. In this case, we have a mother who's a carrier, probably related to British royalty many, many, many generations ago. If she passes on her affected X, her son will have the disease. If she passes on her affected X to her daughter, the daughter is only a carrier. And so notice that her two daughters are not affected, but one of her sons is. All right, so let's talk about pedigrees. Now, real quick, I know I said I was going to move on, but I want to make sure that we cover this. So because women have two X chromosomes, they will always pass on an X to their offspring. However, men will only pass on the X to their offspring 50% of the time. When a mother is a carrier of an X-linked disease, then 50% of her boys will have the disease. But unless the father is affected, 0% of her daughters will have the disease. I want to make that clear. So when we talked about hemophilia, right, we said man without hemophilia has offspring with a carrier woman, then 50% of his sons would have hemophilia and 0% of his daughters. Also, 50% of his daughters would be carriers of the disease. Okay, now let's move on to pedigrees. Okay. So pedigrees are very useful for tracking common recessive disorders within a family or common, genetic diseases within a family. More often than not, it's just genetic diseases. By looking at a pedigree of affected and unaffected individuals, we can get an idea of how these genes and traits are related and also inherited. So this is your typical pedigree. We'll notice that all females are round and all males are square. And you can think about that in real life. Like most males are square and females are a little bit more fluid. So are you a square? Or anyway. All right. So affected individuals. So that means individuals with the disease will have a shaded circle or a shaded square. Sometimes if they're a carrier, like if that you've identified them as a carrier, they'll have a half filled square or a half filled circle. In this case, we're not marking carriers. That's fine. And so we look at this pedigree. And so we see the first generation up here has this Roman numeral 1. And then individuals in that first generation, we label them 1, 2, maybe 3, 4 if there is more. Second generation will have a Roman numeral 2, third generation a 3, and then fourth generation. So I can talk about individuals. I can say, okay, what do you think the genotype is of individual Roman numeral 4, number 4, Roman numeral 4, number 4? What do you think the genotype of that individual is? And we could trace it up and we could look and we say, okay, well, they came from two parents that were unaffected who had an affected offspring. So one of them was likely a carrier. I'm going to guess it was this dude here because he comes from the family that, is expressing this disease pretty often. And so I'm going to say he's a carrier. And if him and her are able to have offspring that are affected, well, then, geez, it's probably she's also a carrier because that would explain the 3-to-1 ratio I'm seeing of unaffected to affected individuals. So I would say, oh, this looks like a recessive disease. It's not affecting males too much more than females in this patterns of inheritance. So I'm not going to immediately say it's X-linked. So that's how we do this pedigree analysis. So we got plenty of practice coming up. So sometimes we'll see dominant versus recessive pedigrees. Now, in a dominant pedigree, all individuals in a dominant pedigree, all affected individuals in a dominant pedigree will have an affected parent. That means in order for you to be affected, you have to have received the allele from one of your parents. And if it's a dominant disease, well, then one of your parents had that dominant allele, so they would have to be affected. And so that's one way that you can identify a dominant versus recessive pedigree. In a dominant pedigree, one of the parents will always be affected of an affected individual. In a recessive pedigree, what we can see is that affected individuals sometimes come from two unaffected parents. And so that means that they are hiding or masking the affected alleles, and then they circumstantially both happen to pass on their affected copy to one of their offsprings, who then is affected. And then you can also notice that none of his offspring are affected, and so that would insinuate that he has a recessive, or this is a recessive disorder. So anytime you have parents that are unaffected that have affected offspring, it's a recessive disorder. Now, when we look at X-linked pedigrees, one of the things that you can look at, and this one is helpful because you can look at the circles and see who's a carrier in this one. And notice the females are the only ones that can be carriers. Notice they're all circles that are half filled. Notice that if you were only looking at affected individuals in this pedigree, the only ones who are actually affected are all males, the square, the square, and the square. And so those are some of the indicators that this is an X-linked trait. All the males are affected, or only males are affected. And anytime that you have an affected female, all of her sons are affected. That would be another identifier. So here's a good question. If a female was affected, what does that say about her father? So another circumstance here, right? The dad can only pass on to a female daughter. He can only pass on his X chromosome. If he was affected and he had the disease on his X chromosome, that means that he had to pass on that to his daughter. So if the female is affected, that means she had to have had an affected father. Okay, so there's some hints and clues and tricks you can use to figuring out what the type of disease is from looking at. All right. So let's do some practice problems. At this point, you're welcome to follow along with me, maybe pull out a piece of paper. I'll give you a chance to pause it and kind of work through it on your own. And then when you're done with this video, when you're done with these practice problems, there's a Mendelian Genetics worksheet that you can work through online. I highly encourage you to do that because a lot of those questions are going to show up on the exam and quiz. All right, moving on. So let's talk about sickle cell anemia for a second. So this is a real thing. This happens in humans. There's a gene, and we're going to oversimplify this, of course. I mean, there's a whole lot of complicated ways to form hemoglobin and stuff like that. But sickle cell anemia is one that follows Mendelian patterns of inheritance. The difference with sickle cell disease is that it's a issue of incomplete dominance. So this is where there's 3 phenotypes for the 3 genotypes. If you remember, complete dominance only has 2 phenotypes. You have the dominant phenotype and the recessive phenotype. For incomplete dominance, there's going to be 3 phenotypes, one for the homozygous dominant, one for the heterozygous, and one for the homozygous recessive. So sickle cell is one that follows that pattern of inheritance. So the big A is going to code for a normally shaped red blood cell. The recessive allele little a is going to code for a sickle-shaped red blood cell. The sickle shape has a the sickle red blood cell has a funny shape, and so it gets trapped in blood vessels. The spleen removes it, which causes anemia. If you're removing a ton of red blood cells, it's removed quickly by the spleen, causes anemia. And then because I said this gene is expressed with incomplete dominance, that means that the heterozygous individuals have these slightly misshapen red blood cells. They're not really problematic unless you go to really high elevations where you need them to bind oxygen extremely well. Sometimes they do get removed by the spleen. So some heterozygous individuals suffer a little bit, but for the most part, they're asymptomatic. You cannot tell if you're a carrier of sickle cell anemia just by looking at you. Yeah, so here's our practice problem. So imagine that there's a family from sub-Saharan Africa that has immigrated to the United States. The mother and father are both healthy, yet several of their offspring end up having sickle cell disease. Well, what are the genotypes of both parents? Let's break this down. Okay. We know that neither the father and the mother have the disease. So that means they could either be homozygous dominant or heterozygous. We know several of their offspring are going to have sickle cell, so that means that they will have to have homozygous recessive genotypes. So let's take a look at this. This individual here, we know this individual would have sickle cell anemia. We know that individual can only be made if the father had a little a allele and the mother also had a little a allele. That's the only way. So we know that the father and the mother have to have at least one little a in their genotypes. We know that they are both unaffected. Both the father and mother do not have sickle cell. So they have to both have a good copy of the big A allele. So the genotypes of the 2 parents are heterozygous. Big A little a, big A little a. All right, practice problem 2. You have asymptomatic carriers, so I'm telling you what their genotypes are, of sickle cell disease, and they end up having offspring together. If they have 12 kids, how many children would you expect to have sickle cell disease? If you know how to do this, or you think you know how, pause it, move on. Okay, so let's figure this out. What are the genotypes of the 2 parents? Well, I told you up here, they're asymptomatic carriers, so they have to be heterozygous. So heterozygous mom, heterozygous dad. What are all the possible sets of alleles they could pass on? Well, let's make our Punnett square. Mom could pass on a big A, or she could pass on a little a. Same thing for dad. So we put those alleles in the borders around our Punnett square. Boop, 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 boop. And then we say, okay, they have 12 kids. How many children would you expect to have sickle cell? Let's take a look. Only 1 out of 4 is going to have sickle cell. If they have 12 kids, well, then we say 25% of their offspring you would expect to have sickle cell anemia. So take 25%, multiply that by 12, and we would say 3 out of the 12 kids might have sickle cell disease. It could be more, it could be less. It's all in chance. Okay, so here's some dihybrid cross practice. So we have a gene that codes for head size, and this is totally stupid. I made this up, okay? So don't be offended. There's a gene that codes for head size that has two alleles, big A and little a. Big A codes for a large head, while little a codes for a small head. The alleles are expressed with incomplete dominance. Pause. What does that mean? That means there's three phenotypes. Big A is going to have a large head. Big A is going to have a medium head. Little a is going to have a small head. There's another gene that codes for hand size that has two alleles, big B and little b. Big B codes for big hands, while little b codes for small hands. Those are also expressed with incomplete dominance. So what does this mean? That means for hand size, you're also going to have three phenotypes. So there's our alleles and what they code for. So what would somebody who is heterozygous for both traits look like? What would their phenotype be? Pause it, see if you can figure it out. They would be normal size head or medium size head. I don't want to go with normal here, but they would have a normal size head and normal size hands, right? Notice I made the picture blend between the two sizes, right, for both. What would someone look like who was homozygous dominant for head size, but homozygous recessive for hand size look like? Go ahead and pause it, see if you can figure that out. Okay, so this individual, oh, that's for the last individual. So for this individual, they're going to have a big head. So they'll have big A, that codes for a big head. And they're going to have little a, which would code for, am I doing that right? Sorry. Big A for a big head. Little b for the small hands. And then finally, the last one, if somebody had a, well, there's your genotype and there's your phenotype. If someone had a small head and medium hands, what would be their genotype? So pause it, see if you can figure out the genotype for that phenotype. A small head, that would mean little a. Medium hands would be the intermediate hybrid, big B, little b. So little a, big B, little b. Right. And then, all right, let's do a practice with our Punnett square. So here we go. A man with a large head and medium hands has offspring with a woman who has a medium head and small hands. What percentage of their offspring would you predict to have large heads and medium hands? That sounds like I mean, it just sounds like, where do you even start? Start with the genotypes of the parents. Okay. Because there are three phenotypes for the three genotypes, you know what the parents' genotypes are just by the description of their phenotype. A man with a large head, what would that genotype be? Big A. Medium hands, what would that genotype be? Big B, little b. So the man's genotype is big A, big B, little b. He has offspring with a woman who has a medium head and small hands. So what would her genotype be for head? Big A, little a. Her genotype for small hands? Little b. What percentage of their offspring would you predict to have big A, and big B, little b? Let's find out. So we figure out what the two parent genotypes are. We already said the man was big A, big B, little b. We already said the woman was big A, little b. So what are all the possible combinations of these alleles that can go into sperm and egg? Well, the man can only pass on a big A. But due to the law of independent assortment, he could pass on the big A and the big B, or he could pass on the big A and the little b. So those are the two haploid sperm combinations that he could potentially make. The woman, the same thing. So she could pass on a big A, but she can only pass on a little b. So she could either pass on a big A and a little b, or a little a and a little b. And those are all of the possible sperm and egg combinations they can make. Now we take those and we're going to place them into a Punnett square. So the man's sperm, one of his sperm is going to be a big A, big B. One of his other sperm is going to be a big A, little b. The woman, the same thing. She's going to have a big A, little b, and a little a, big B. And now we ask the question, what percentage of their offsprings would have the genotype big A, big B, little b? And so we fill out our Punnett square. And we say, okay, what percentage is going to have the genotype big A, big B, little b? 25% will have large heads with medium hands. All right, let's do our practice pedigree problem. I asked the question, is this pedigree autosomal or X-linked? Well, let's see, is the disease dominant or recessive? What is the genotype of big B? And what's the genotype of individual C? Well, let's figure it out, right? If do we see any, let's first ask the question, is it recessive or dominant? Are there any individuals that are affected that have unaffected parents? No, they all have unaffected parents. So it's very possible it's dominant. It's very possible it's dominant. This individual has affected offspring. This individual has an affected offspring. This individual has an affected offspring. One thing you do know is that if it is dominant, the individuals that are making offspring are only heterozygous, right? If this individual right here, this affected individual was homozygous dominant, every single one of their offspring would be affected because they could only pass on the dominant disease. So we know these individuals are heterozygous. This disease is likely dominant. Okay. Now, is this X-linked or autosomal? Well, let's figure this out, right? Are there more affected males than females in this pedigree? No, they're about the same, 3 and 4. Okay, so that's my first level of analysis. My second level is this. If it was X-linked, all of the male offspring from an affected female will also be affected. Is that true? No, because here's a male right here that's not affected from his affected mother. So we know it's not X-linked. We know it's probably dominant. And so if I was to guess the genotype of this individual here, C, I would say, well, that genotype, since it's unaffected and it's likely a dominant disease, they're probably homozygous recessive. All right, so finally, so that's individual C. Now let's ask for individual B, right? What would be his genotype? We figured out hers was a homozygous recessive, which is fine. What is this individual's genotype? Well, his mom likely passed on a bad copy of the dominant disease to him. If he was homozygous dominant, right, that means that his dad would have had to have the disease, so that can't be true. So he has to be heterozygous. Because his dad's unaffected, it's a dominant trait. We're watching the transmission of a dominant disease. His dad's unaffected, so that means his dad has to be homozygous, recessive for it. Mom is heterozygous because only half of her kids are affected. Mom is heterozygous, so mom likely passed on her dominant bad allele, and dad likely passed on his recessive good allele. And so that's the individual's genotype. He's male and he's heterozygous. Female, homozygous recessive. The disease is most likely a dominant disease. All right, another practice pedigree. These are always fun to do. Is it autosomal or X-linked? Well, let's take a look. Are there any affected females? I don't see any affected females. Are all the males affected? Ryder, hold on, buddy. Okay, so we were looking at this pedigree and we said, okay, are any females affected? No. Are there more males and females that are affected in this pedigree? Definitely. Only males are affected, right? All the squares are filled in. So it's probably X-linked. Is it dominant or recessive? Well, let's look at the affected individuals. If the affected individuals have parents that are unaffected, well, then it's likely recessive. If it was dominant, every individual that's affected would have to have a parent that was also affected. So X-linked, probably, and recessive. What's the genotype of individual 2-1? So this is where I'm giving you that reference. 2-1. Well, it's a female. It's X-linked. She had an unaffected father, so she's probably okay. So she has at least one good X chromosome. Her other X chromosome came from mom. Whoop, right? And then so she has an X and an X. One X from dad, one X from mom. We know mom has a bad copy of an X chromosome that she gave to her son right here that's affected. So mom is definitely a carrier. The question is this female also a carrier? Now, the way to figure that out is to go down the line and say, okay, what are the genotypes of her offspring? Okay, well, she had a female daughter who married a male that was unaffected and they had a male son who was affected. So this male has to have a mother who was a carrier. And if she's a carrier, that means she likely got her bad copy from her mom or her dad. It could come from here too. And so I would say this individual is most likely heterozygous. She has an X with like a capital H and an X with a lowercase h. So heterozygous for that individual. And then let's come down here for this individual, number 4-1. They're an affected male. So we know their genotype is mostly, it's going to be XY. But because they're affected and they only have one copy of the X, then their genotype has to be X with like a little h and then a Y. So that would be that. And that is the end of those practice problems. Hope you guys enjoyed it. I will see you for the next lecture.